everyone for joining us for the June 2024 seminar on physical genomics. My name is Benjamin Keene here at CPGE at Northwestern University. We host this seminar every month. We're very happy and excited to welcome our invited speaker today, Dr. Michael Elowitz. He is a Howard Hughes Medical Institute investigator and the Roscoe Gilkey Dickinson Professor of Biology and Biological Engineering at California Institute of Technology. Dr. Elowitz's lab brings synthetic approaches to understanding and programming multicellular functions, including multistability, cell-cell communication, epigenetic memory, and cell fate control, and provides foundations for using biological circuits as therapeutic devices. Dr. Elowitz received his PhD in physics from Princeton University and did postdoctoral research at Rockefeller University. He's been recognized with the MacArthur Fellowship, the HFSP Nakasone Award, Presidential Early Career Award, is an Allen Distinguished Investigator, and has been recognized with the American Academy of Arts and Sciences and election to the National Academy of Sciences. So we have a very distinguished guest today. Dr. Elowitz, it's an honor to have you here with us. For our audience, a quick word. You can type questions at any time into the Zoom chat box, and we'll address them at the end of the talk during the Q&A session. Or you can just raise your hand at the end and ask. That works as well. If you'd like to learn more about the research and training programs we have here at Center for Physical Genomics, you can visit us at physicalgenomics.northwestern.edu on the web, or follow us on our YouTube channel, our Twitter X account, or on Instagram. At this point, Dr. Elowitz, if you'd like to share your screen, I can turn the floor over to you and we can get started. Thank you so much once again.
Wonderful. Thank you very much for inviting me. It's really fun to be here.
Let me unshare my screen.
Oh, yeah, I think that's...
Okay.
Okay, let me try that. And let's see if that works. How does that look? Okay.
That looks great.
Okay. So, all right. So I thought what I would try to do is tell you, I kind of structured this a little bit like a YouTube video in the sense that I'm going to tell you about some work we've been doing in the lab to try to understand principles of circuit design in multicellular systems. And I'm going to try to have a few little ads for some systems and techniques that I think are broadly useful at the beginning, and then kind of focus on one main idea. And that idea is encapsulated in this title. It's the idea that in biology, we have a lot of systems that have families of proteins that interact with each other in a many-to-many way, and that those systems are really useful for multicellular behavior specifically. So I'll hopefully explain all of this as we go. Okay, so here we go. I'm just talking to the screen. So, okay, here we go. So I always like to start by just reminding myself and everyone, I guess, that the cell is kind of an amazing little device. It can proliferate, differentiate, communicate with other cells, coordinate with them to form, complex patterns, tissues, organs, develop into multicellular organisms, and then even generate immune systems that patrol those organisms and eliminate disease or heal injuries. So this is kind of an amazing device. And when we kind of zoom in on a lot of these incredible functions, what we find is that they are really carried out by circuits, molecular circuits, and often cellular circuits of interacting components or interacting cell types. And in my lab, what we've been doing for a number of years is kind of trying to take different functions and try to understand the principles by which they are implemented by these different circuits. So this is a little gallery of some of those, some of the kinds of systems that we've been looking at, a couple of which I'm going to talk about today. As we've been doing this, we've also been, so when we think about these circuits, we try to ask a few basic questions. What is it that a given circuit does for the cell functionally? Why does it use one particular design instead of some other design? What's special about the designs that we find in nature? And what designs are good if we want to build circuits that provide these functions synthetically? And more generally, just like how can we actually start to not just analyze the kinds of behaviors I just showed you in natural systems, but also start to program them or create them from scratch. That's the synthetic biology aspect. So these are sort of our general motivating questions. And alongside of that, we've also been developing a variety of tools that can kind of both enable us to, and hopefully the community, to better understand circuits and manipulate them in new ways, or provide useful circuit level functions for therapeutics. So I thought I would first tell you about 3 of these tools, just very, very briefly, in the hopes that people on this call might find one or more of them useful. And I'll just say that if they do, feel free to contact me and hopefully we can help make them useful to you. So the first one comes from a general problem that we have in a lot of multicellular systems, which is that we can take a snapshot of cells, if you're using single-cell RNA sequencing or some of the imaging techniques like seqFISH or MERFISH, and we can see the states that cells are in now. But to understand how they got there, we would really like to know something about their history. One thing we'd like to know is how they're related to each other. What are the lineage relationships? What's the lineage tree that gave rise to these cells? And over the last number of years, several groups, including ours, have been developing strategies to reconstruct those lineage trees based on the same principles that we use for phylogenetic inference, but taking advantage of engineered systems that can introduce stochastic mutations into barcodes that have been engineered into the genome. And by putting in random mutations as cells, or by engineering the system so that these barcodes randomly mutate continuously in time as the cells grow and divide, each cell can accumulate a different pattern of mutations. And by reading those out, we can reconstruct the lineage trees of all of the cells. And we've been working on techniques for a number of years that allow you to do that with imaging. So we'll read out the mutations directly within the cell, within images of the cells. And sorry about that. Okay. And so these are just some examples. We just put a paper on bioRxiv this year, which shows how we have a new system using base editors that can allow us to reconstruct the lineages of, in this case, mouse embryonic stem cell colonies, but the system could be used really for any cell type of interest. And so what you're seeing here is just an example where this is a colony and we can kind of see the lineage relationships among all the cells, but also the states that those cells are in using sequential single molecule FISH. Okay, so that's one technique. A second thing we've been interested in is the problem that even with that base MEMOIR technique and with single-cell RNA sequencing, these are very powerful techniques, but they all require killing the cells in order to analyze them. It would be great if you could reconstruct the longitudinal dynamics of cells or cell populations over time without having to kill them. And so Felix Horns, who's a fantastic postdoc, developed a system in the lab which we call Courier, which is an engineered RNA exporter. And what it does is it assembles, it's based on self-assembling engineered nanocages, and Felix modified them so they specifically and efficiently package just an RNA of interest and then export it in an extracellular vesicle. And by recovering those extracellular vesicles and sequencing them without killing the cells, just recovering them from the media, you can kind of keep track of how many copies of each barcode are present. And you can use that as a proxy for the population sizes of cells of different types. So you can really monitor the dynamics of different cell populations non-destructively over time. The other thing you can do with this system, it's really totally different, but it's a kind of 2 birds with 1 stone kind of thing, is you can use these engineered exporter cells to deliver RNA from one cell to another. And if that RNA is an mRNA, it can be expressed in the receiving cell. Now you have a way, different kind of delivery system for mRNA in which you can start to engineer cells that could go and bring mRNA to other cells, which would be useful for either making systems that kill other cells or reprogram other cells or edit other cells, but do it all in vivo. So this is a second system. This was published last year. And the third of these little advertisements I wanted to provide here is has to do with the problem of gene therapy, which is that even though there's been an enormous amount of progress in terms of delivering DNA to cells for for doing gene therapy, it's still difficult to control. So people often package a therapeutic DNA in an AAV capsid. But first of all, you have to get the capsids to the right organs. For example, if you're trying to target the CNS, but you don't want to target the liver. Then different cells within a tissue may take up different amounts of the virus and therefore have different levels of expression of the therapeutic gene. And sometimes that can be kind of more systematic, or sometimes it can just reflect Poisson variation in uptake. So there's many ways in which it's very hard to control for every cell exactly how many copies of the gene it gets. You can think of this as like the chocolate chip cookie problem, that it's hard to control exactly how many chocolate chips land in each cookie. But there is a circuit level solution for this, which is an incoherent feed-forward loop. That's one of the circuit motifs that Uri Alon discovered many years ago. And other people have shown that in the right regime, this kind of a circuit can give you a sort of gene dosage independent expression, in which the expression level of the gene becomes at high enough gene dosage independent of how many copies of the gene get delivered. So this is a great property for gene therapy, especially if you could tune what that set point is. And so we recently, this is work from a grad student, Mike Flynn, another grad student, Ron Du, engineered a set of circuits that we call dimmer circuits that co-express a microRNA that self-targets the gene of interest. And in these systems, the gene makes an mRNA, which makes a protein, but the same construct also delivers a microRNA that downregulates the same mRNA. And to make a long story short, with a lot of engineering, we've been able to get this into regimes in which you can get a pretty wide range of gene dosages producing a constant or a constant level of expression of the target gene. So this we now have put into a mouse model of a very dosage sensitive gene therapy candidate disease called Rett syndrome in mice. And it seems to be providing benefits compared to regular unregulated gene therapy. So this is a collaboration with my colleague Viviana Gradinaru and Acacia Mayfield, who's a great student in her lab. Okay, and so this is also, these papers are online now in bioRxiv this year. So anyway, I hope these constructs will be useful to people doing a variety of different kinds of things. Okay, so with that, that's the end of the ad part. Now I just want to talk about the main idea for the talk. And I think this starts by, in synthetic biology, for the most part, most of the field has been focused on engineering behaviors in individual cells, kind of focused on the level of one cell and what we can get that cell to do, whether it's manufacture useful compounds or materials, or provide certain kinds of functional behaviors or something else. But there was this great transition in the history of life from single cell level to the multicellular level, where you go from kind of generalists to specialists, like specialist cells that can interact with each other to carry out the different functions necessary in a multicellular organism. And in our lab, we're sort of interested in the circuits. And so one question is like, what changed in circuit design to enable multicellularity? This is not a rigorous statement. It's kind of just an impression. But one thing you notice is that when you look at bacteria, you see a lot of examples of very strong and specific interactions that are kind of one-to-one. The classic example of this are the two-component signaling systems. And Mike Laub famously showed that in those systems, you have histidine kinases that phosphorylate cognate response regulators. And each histidine kinase kind of phosphorylates one of those response regulators, and there's very little crosstalk between them. By contrast, when we go to mammalian cells or to multicellular organisms, we often see signaling systems that look very different, in which you have a set of components like ligands that interact in a many-to-many way with another set of components like receptors. And so you see this at many different levels. One level is, like I just said, families of ligands interacting with families of receptors. We see that with BMP, TGF-beta, Wnt, Notch, FGF, many pathways. One level down, we see something similar when we think about how those receptors interact with effectors. So for example, GPCRs a variety of GPCRs can interact in a many-to-many way with different G proteins. Going further down, we also see it with the transcription factors in the cell, which often come in families, such as the basic helix, loop helix family, or AP1, or others. And transcription factor proteins in those families often dimerize in a many-to-many way with other members of the family, and those dimers can carry out different functions. We see something similar again in adhesion. So when you look across all of these different systems, and you come to those systems with the goal of trying to really analyze and understand them, this situation can be initially heartbreaking, because it means that to understand any of these systems, you really are going to need to know a lot of different parameters. There's just so many different interactions to worry about. So one question is kind of why are these architectures so prevalent, in particular in the multicellular organisms? Is it an evolutionary accident of the way the networks evolve, or do they carry out, or does this architecture provide some specific function? And if it does, what is it? Okay. So one thing you might think, just looking at these diagrams, is they're superficially similar to diagrams of neural networks, and that's kind of suggestive of the possibility that maybe they're performing some kind of computation. Now, an interesting aspect of these computations is that in the multicellular context, different cell types may be expressing different combinations of the components of a given pathway. So, for example, these three cells are expressing different receptors in each of the three cells and different transcription factors. And so it's possible that there really isn't just one network providing one function or one computation, but actually different versions of that network expressed by different cells computing different things. So if you think about this, there's sort of what I just sketched out is there's many layers of these many-to-many interactions, from ligands to receptors, receptors to transcription factors, and transcription factors among themselves. And we've been very interested in just trying to understand the computational capacities of these systems. And so today what I'll try and do is talk about three aspects of that. First, when you do have these systems, how are the components deployed or expressed across all the different cell types? What kinds of computations can they actually perform? And how do they enable the higher level properties of multicellularity? So to address the first question about expression, this is work from Alejandro Granados, who was a postdoc in the lab and initiated this project. And he worked very closely with an amazing undergraduate, Niv Kanrar. And this project has also been joined by a really spectacular new postdoc, Judy Schoen, more recently. Okay, and the starting point for this was just thinking about if I have a pathway, which is really a family. Okay, pathways could mean different things to different people. Here I'm going to think about a family of related proteins in the same pathway. For example, all of the different receptor subunits of the TGF-beta pathway. And if I imagine that I have a group of different cell types that are related to each other in terms of their overall similarity, maybe immune cells here and fibroblasts here and neurons here, I can ask, how do these different cell types vary in their expression of those, in this case, receptors? So one thing you might expect is that cells that are overall more similar to each other also exhibit similar patterns of expression of these different receptors within the pathway. In other words, the similarity among the pathway profiles follows the similarity of the overall expression profiles across the entire transcriptome. On the other hand, it's also possible that you would get something very different, in which maybe cells that are closely related to each other could have very different profiles, but cells that are very distantly related to each other, like the ones here in this indicated by these arrows, could actually have the same profile. This kind of behavior suggests that there are profiles that are recurring across different cell types and are dispersed across cell types that are very different from each other. We call that a pathway motif. It's kind of a recurrent profile of the genes within the pathway. So we set out to kind of see what these distributions, which way does it look? Like this, or like something else? And this was made possible because of all of these amazing cell atlas data sets that different groups have been developing. And so if you just take those single cell atlas data sets and you cluster cells that seem to be in the same state, you can make pseudo-bulk cluster average gene expression profiles. So each of the profiles represents one cluster of many individual cells. And then we can sort of see what they look like. So in this plot, what you're seeing is each of these very thin lines represents one of those cell type clusters, not a single cell, but a cluster of related cell types. And each of the rows represents one of the TGF-beta receptor subunits. So when you look at this, you can immediately see that the expression profiles are kind of complex. So each receptor subunit is expressed in multiple, multiple cell clusters and profiles. And then each of those profiles usually contains expression of multiple receptors. So it's very combinatorial. But you can also see that there's groups of cell types that really seem to have very similar expression profiles. Now we can ask, when you have multiple cell types that express the same profile TGF-beta receptors, are they doing that because they're very similar to each other overall, like I showed in the first of those two cartoons, or are they dispersed? So some of them are quite dispersed, and the examples I'm showing here are examples of that. So here the gray represents a UMAP plot of the whole transcriptome. Okay, so you're just seeing kind of all the different cell types in the organism. And I should say this data set, in this case, and we've done it on different data sets, but this is the human tabula sapiens data set developed at Stanford. And I've lit up here in different colors cell types or cell clusters that express each of these 5 different profiles. And what you can kind of see in these plots, and can be made more rigorous with other techniques, is that in general, each of these 5 profiles tends to be spread out among a variety of different, not identical cell types. Now, I want to say that not everything looks like this. We also have the opposite behavior, in which sometimes you have TGF-beta receptor profiles that are really confined to really one set of closely related cell types. So that's what's shown in these examples on the bottom row. We call these the private profiles, and we call the ones on the top the motifs, and both exist. I want to focus on the motifs because I think they're particularly interesting, since they deviate from what you would expect from a more hierarchical view of transcriptome expression. So on the left here, what you see are the different motifs. So each row represents one of them. And on the right, you see how many cell types in a given organ, which is the columns here in the pink, express each of those different motifs. And once again, what I think you can see is that it's very combinatorial at this next level. Each motif is expressed in multiple organs, and most organs include cell types that express multiple motifs. So this is pretty interesting. It means that you could have 2 cell types sitting in really different cell types altogether and are existing in different organs, but have somehow converged to use the same TGF-beta receptor profile. Now, you might ask, is this a weird thing about TGF-beta, or is it more general? And we started doing this analysis on other pathways. So for example, if we look at the nuclear hormone receptors, just as one example, you can see a qualitatively similar pattern in which you have a variety of different recurring motifs shown on the left. And then again, each of those motifs seems to appear in multiple organs, and each organ seems to contain multiple motifs. Now, the other thing that's interesting is that even though they're qualitatively similar, that's not because you have correlations in the usage of motifs between different cell types. So it seems like different, the choice for a given cell type of which motif it's going to use for one pathway appears to be pretty independent of the choice of what motif is going to be expressed in a different pathway. And you can see that here just from a sort of mutual information metric that we use to quantify those correlations. And the dark colors here indicate that most of the pathways are pretty weakly, if at all, correlated in terms of their motif with the motifs used by other pathways. So all of this suggests, I think, something a little bit surprising, which is that when we think about the organization of a cell type, we can kind of think of it as a little bit of a prefix menu in which most cell types are using most of these pathways most of the time, but they're operating them in different modes, these different motifs. And the choice is sort of independent. So I like to think of it as a kind of prefix menu for the cells in which every cell type, I should say, is ordering its own particular combination of the appetizer, the entree, and the dessert, which here means which mode each of these different pathways is operating in. Okay, so another way to say that is that the pathway expression is mosaic among cell types. This is also interesting because it's less complex than things could have been if as cell types became overall more different from each other, they also made every pathway become more different from that the expression profile of that pathway and other cell types. If the pathway expression profile is diverged along with the overall transcriptome profiles, then there might have been a much larger space of possible pathway profiles to worry about or to think about. So this is a simplification. Okay, having said that, now that we know these profiles exist, we don't automatically know how they differ functionally, if at all. Maybe these different profiles really all do the same thing, or maybe they correspond to different functions in the cell. And to think about that, I want to take a step back to some work we did a few years ago, looking at one particular signaling pathway called BMP, or bone morphogenetic protein. So BMPs, it has bone in the name, but they're not really bone specific. What's interesting about them is they consist of a family of ligands that interacts in a many-to-many way with a family of receptors. There's actually hundreds of different receptor complexes that can be formed out of different combinations of the TGF-beta or BMP receptor subunits. And then these activated ligand receptor complexes can phosphorylate effectors called SMADs that activate target genes. And Yaron and Tabi, who's now at Weitzman running his own lab, when he was a postdoc in the lab, together with James Linton in my lab, they discovered that if we titrated the concentrations of two different ligands simultaneously, sometimes they would combine in a boring additive way, like you see here with BMP4 and BMP9, but sometimes you'd see things that represented computations, for example, even simple ones. Here, you can see that GDF5, which is a BMP ligand, inhibits activation by BMP4 in a dose-dependent way, resulting in an output that's proportional to the ratio of the two ligand concentrations. Or on the right here, you see an example in which either ligand activates, but together they produce a smaller response than either one alone. Call that an imbalance detector. And we were able to explain some of these behaviors in a simple model in which different ligands and receptors compete to form various complexes. And by competing to form those complexes, and by having the complexes have different signaling activities, you can explain a lot of the kinds of behaviors that we see. Now, that just says that the pathway can perform some simple computations. But you might also want to know why, or to what extent does that pathway always perform the same computation on the same ligands? It turns out that when you change the expression of the different receptors, you also change the computation that's performed. So you can see that here. This is one of the cell lines we like to use to analyze the BMP pathway. And then these three heat maps are the same pair of ligands. They're the activity of the pathway as a function of the same pair of ligands. But in cells where you've neither knocked down or overexpressed specific receptor subunits, or in a different cell type altogether with a different profile of receptors, in this case, mouse embryonic stem cells. And what you can notice here is that cells with different receptor profiles respond to different ligand combinations. So this is a functional aspect of the receptor profile, is that it seems to control how you respond to different ligands and which ligands you respond to. If we turn that statement around, we could suggest that maybe what this is doing for the system is allowing it to use combinations of ligands to selectively activate particular cell types, the ones that are more responsive to that combination. And you can see that here, that if you just look at, in a very simplified example, these different points in the space of concentrations of two ligands, then each of those points kind of differentially activates different combinations of cell types. So you can think of this as a kind of addressing system in which different cell types can be selectively activated by different ligand combinations. Okay, so that's what got us into this idea that perhaps these many-to-many systems are really computing, and that they're computing different functions in different cell types, that there's a contextual effect there. Now I want to step down one level to the transcription factors, which in some respects are even simpler. So when you look at families of transcription factors, you find that they often function as dimers, so either homodimers or heterodimers. Here's an example from the nuclear receptor family, and the arrows here represent the known dimerization interactions among these components. But there's many other examples as well. And about 30 years ago, my colleague Barbara Wold, thinking about one of these families, which was the basic helix loop helix family, which is important in myogenesis, she wrote a paper which in the discussion section kind of suggests that these dimerization networks could be computing interesting functions. So I'll just read one little part here where she points out the differential dimerization among these components provides an attractive mechanism by which changes in cellular phenotype can be initiated by altering one or a few components. So what she has in mind here is the idea that I'm going to talk about now, that these circuits act as little computers, and by perturbing one or another component, that perturbs all the other components, and you can end up having a complex response to a particular input. Okay. On the other hand, dimerization itself is a pretty boring interaction in the sense that it's just two proteins sticking together, and it's not catalytic, it doesn't consume ATP. It's almost one of the most boring things you can imagine proteins doing. So it's not obvious that a simple network of interacting proteins could do anything interesting. And so we've been trying to understand how powerful are simple dimerization networks of the type that appear so often in nature. And this is a project that was really led by Jacob Pars-Gold with a fantastic collaboration with Ben Emmert, Matt Levine, who was a student with Andrew Stewart and has recently moved to the Broad. Okay, and the way we think about this system is we say, okay, there's a handful of different protein species, and they can dimerize with each other to form homo or heterodimers. And there's some affinity for each possible dimerization, which we can call Kij. And there's also some conservation of mass in the sense that the total amount of any of these proteins is conserved. So we can represent these networks by, sorry about that. We can represent these networks graphically by saying that by drawing them this way, where here each protein is represented by one of these circles. The size of the circle represents the abundance of the protein in the cell. Different cell types could express different amounts of the protein, and the size of the arrow represents the affinity for that particular dimerization interaction, either homo or hetero. Now, if we want to understand what computations are possible, we have to think about two different ways the networks could vary. First of all, you could have just two different networks with different affinity patterns, as you see here, or you could have the same network with the same affinities, but just change the expression levels of the components. That corresponds to what would happen in different cell types that express different levels of these proteins. And you can get a range of different computations either way. So we can think of the overall set as kind of the expressivity in analogy to the terminology in the neural network field. And we can think of more narrowly the variation in function you get just by changing the protein expression levels as what we call versatility. So just to show you that these systems can be powerful and can do interesting things, I'm going to start with some very simple examples. So here's one of the simplest things you can do is imagine I just have this protein M2, and I'm going to say the output is the homodimer of that second monomer, so the orange homodimer. And now if I start adding as an input a second protein, this green one, that can dimerize with the orange one, and maybe very strongly, it's going to pull or sequester orange proteins away from homodimerization. So we can turn off the concentration of the orange homodimer. So this is a simple off switch, very, very simple. If I add one more component, I can flip the off switch to an on switch by having the green protein pull orange monomers away, the orange monomers pull blue monomers away from the blue homodimer, and now I can flip it around. If I put those two together, I can get a bandpass function, or if I add one more component, I can get a kind of negative bandpass here. So that's with one-dimensional responses. But what's cool about these systems is that they can also function as multi-input and even multi-output signal processing systems. So for example, here's a 4-protein system that can compute an A and not B function of 2 input proteins. Here's like a NOR gate. Here's a double bandpass that kind of turns on only within a window of 2 different input concentrations. Or here's an XOR, which is a more complex type of response. So I think what you can see here is that as the complexity of the circuit grows, the complexity of the functions also increases. And you can start to get a feeling for the diversity of functions that these simple dimerization networks can compute. Now, what about the versatility? So what you can see in this movie is if we vary the proteins that are neither inputs nor outputs, what we call the accessories, that are the bystanders there in the network, that can actually, if you take the one case here, like this double bandpass, move around where the double bandpass is in space, or it can qualitatively change a function from, in this case, an and to a nor, or to other functions as well. So that shows that these networks can be versatile functionally as well as expressive. It also raises the question whether it's very hard or difficult or rare to find networks that can have this ability to both be expressive and versatile. How hard is that? Do you have to fine-tune all of the affinities to get that to work? So we tried to ask that question computationally, and we did it by optimizing, by kind of tabulating a set of functions and then making random networks and optimizing the expression levels of those random networks to see for each network how many functions we could get it to perform. And the result is here in this plot. So here the x-axis is the size of the network, the number of different protein species in it, and the y-axis is the fraction of all possible functions that could be computed by some network of that size that are computed by a particular network in question. So each black dot represents one random network. Again, we've not changed the affinities here at all systematically. And I think what you can see is that when the network size gets large enough, like above 6, then you start to see that most of these black dots are pretty high, which means most networks can compute most functions. I think what this is telling us is that these networks don't necessarily need fine-tuning in order to be functionally versatile. And if you think about replaying the tape of evolution in the Stephen Jay Gould sense, it suggests that maybe we would always get dimerization networks, but the particular values of the affinities might be very different because there are many solutions. Okay, so far I've told you that these many-to-many systems have some interesting structure in terms of their expression. They're expressed in motifs, and that they allow something special, which is computation that can be tailored to different cells, to form different computations in different cell types by varying expression levels of components. Now I want to say, but all of that has been focused on a feed-forward analysis where we just think of these systems as input-output systems. But a lot of them in nature are involved in feedback loops. And we see them appearing in systems that have, that control cell states or cell types. And so in the last part of the talk, I just wanted to add feedback to the story and try to think about what role they could play in controlling cell fates. And the starting point for this is to just think about the amazing result that starting from pluripotent stem cells, many researchers have discovered protocols or patterns or sequences of perturbations to key signaling pathways or other things that can direct cells or steer them into a huge variety of different states. Now, which states can exist, which possible transcriptome profiles are actually stable states, is in principle determined by underlying regulatory circuits. This is an example of the myogenic circuit that controls muscle cell generation. But for the most part, even though we actually know quite a bit about how to push cells into particular states, we know, in my opinion, a lot less about the underlying networks that explain why those states exist and which states can differentiate into which other states. Those circuits have to have a few interesting properties. They have to be able to enable the sorts of hierarchical or progressive differentiation that you see in natural systems. They have to allow control of cell fate by signaling pathways. And at a higher level or an evolutionary level, they have to allow something that we see in nature, which is that as organismal complexity increases, the number of cell types also grows, and many of those cell types are using some of the same sets of components, but now just generating more states. So there's some kind of expandability during evolution. One framework for thinking about how you get all of these different cell types is the idea of multistability. And I think this goes back at least to Max Delbruck. In 1949, he made this little sketch of two metabolic pathways that were cross-inhibiting each other and could, in principle, be bistable, so that one or the other, but not both, could be active at a given time. And he suggested that, this type of model could account for multiple steady states in different cells, similar to the phenomenon of differentiation. We can make this framework a little more mathematical by thinking of it as a dynamical system, like dx dt is a function, the velocity of the system in a phase space is just a function of its position, plus the influence of external signals and noise. So in this picture, like in this toy example here, I have two black dots. Those represent stable attractors. And they have the appealing feature that if I were to perturb away from them because they're stable, the cells would come right back. But if I were to overexpress one transcription factor, like here, crossing this threshold by expressing a lot of A, then the dynamics of the circuit would drive me not to stay in some weird state here with a lot of A, but actually drive me into a different attractor that represents a real cell state. So this is an attractive idea, to me anyway, that we could explain cell states in terms of multistability, but I don't think we can tell just from these toy, just what we can draw on, or do on the computer or write on a piece of paper, whether this sort of thing is really possible in cells where you have so much more noise and all kinds of other unknown variables. And so to address that question, I had this incredible student who's now moved on to his postdoc, Ron Zhu. And the question that he asked is it possible that we could actually build from scratch our own cell fate control system? How would we do that? What would it take to do that? And of course, initial question is, what design would you use? How would you build such a system? And this is inspired by a true story in the sense that what Ron did is he looked at some of the best studied circuits to try to kind of figure out some key features or principles in many of their designs. And what you see in many of these cell fate control systems is that there are families of transcription factors that, as I just was talking about, dimerize in different combinations. So for example, MyoD is a master regulator for myogenesis. It can heterodimerize with another factor called E47. They can activate together myogenic genes. But MyoD can also interact with another protein called ID, or inhibitor of differentiation, to make an inactive complex. And then ID can effectively sequester MyoD away from E47, giving you or inhibiting the differentiation. So in thinking about that, Ron came up with this design, Multifate 2, which represents, I think, one of the simplest circuits that you could imagine supporting multistability, as I'll show you in a minute. So let me say what the circuit is. So there's two genes here. Each one can homodimerize, and the homodimer, but not the monomer, can activate more expression of the same transcription factor. That's true for A and B. But just like MyoD and the ID genes, or ID protein, the two proteins can also bind to each other and form an inactive complex. And when you analyze this mathematically, you can separately do the same exact kind of protein dimerization analysis I showed in those dimerization networks, and that happens at fast timescales and therefore can be treated as equilibrium. And at slower timescales, we have a second process of autoregulation of the circuit components. So in some parameter, the behavior of the system depends on the quantitative parameters of all of the different interactions. But here I'll focus on one parameter regime where you have, for example, here, two factors, three attractors. Those are the black dots, and those represent stable, steady states of the system. Now, so in other words, just two of these proteins in multifate can give you tri-stability with three states in a model. So to see if that really works in real life, Ron first built some components that could carry out this, or implement the circuit. And he had to make sure they would have this property of activating only when they're homodimers, and also allowing external control. So to do that, as transcription factors, we started with these engineered zinc fingers, which are a nice modular DNA binding protein system that's been developed by many labs, including most recently Mo Khalil's lab. And we put activation domains on them. And when you do that, each of the zinc fingers here with three fingers recognizes a 9-base pair of site, which is really too weak or too non-specific for much activation. But if you attach dimerization domains, then they stick together and they can collectively recognize a double length site of more like 18 bases. And so what we see is that in this system, the homodimers activate effectively, the monomers not so much. Okay, having said that, the activation by the monomers is not zero, it's low, but not zero. And so Ron improved that by engineering some arginine to alanine mutations in the DNA binding domain. And what that did is it reduced the monomeric activity while preserving the dimeric activity. The other thing he did is he put a lot of gadgets on the system, so fluorescent proteins so we could read out what's going on, a drug-dependent dimerization domain so we can control the strength of dimerization with AP1903, the drug, and a degradation domain so we can control the stability of these proteins with a different drug called trimethoprim. So once we had these, we could integrate them into cells, in this case, CHOK1 cells. Each clone we get, because we've integrated it using a system called piggyback, a transposon, gets a different number and distribution of these different components. I'm going to focus here again on this symmetric tristable regime, where if we leave out the drugs and the proteins are unstable and not dimerizing, all the cells are off for both of the two genes shown in this flow cytometry plot. But if I add those drugs, then the circuits go to one of three different states, as you can see here, and that's very reminiscent of the tristable regime I just showed you. Now, to see if the states are really stable, we sorted them out and then cultured them for, in this case, more than a few weeks, or more, well, 18 days. And what you can see is that the cells that are sorted in one population tend to stay in that population, suggesting that the states are stable. On the other hand, you might worry that they're too stable, and maybe what's happening is they're getting stuck through some type of chromatin-based epigenetic mechanism. And I'll just tell you, I'm not going to show you, but I'll just tell you that that's not the case, and we can still perturb them away from these states by expressing transcription factors. Okay, so far we saw with two genes we can get tristability, but the nice aspect, the really nice thing about this circuit is that adding a third gene without re-engineering anything else can expand the number of states that you can get. So here what I'm showing is the 3-gene circuit in the same parameter regime that we looked at before, and you can see that it generates 7 different states. And now you can see those, that all 7 are stable, and I'll show you this in a movie. Here, each of the 7 states can be visualized in a different color based on the combination of transcription factors that are present. And what you can see is that the cells are growing into colonies and remembering what, and all the progeny are the same color. So this says that each of these 7 states can actually be stable. Most recently, another student, Evan, in the lab, actually added a 4th transcription factor and showed that we could get 15 states consistent with model predictions. So if we look in the model, we can see that there's exponential growth in terms of the number of stable attractors with the number of transcription factors, but only up to a point. So enough to get like hundreds of different states, but eventually it saturates, and that's because only the homodimers, which grow as n, are active. The heterodimers grow as n squared and are really inactive, not doing anything. And so as you get to larger numbers of species, you start to lose these higher order states. Okay, I see that we're coming up on the hour, and I want to leave time for questions. So I'll also just briefly tell you that this circuit can do other stuff, like progressively differentiate into multiple different fates, but I'm going to skip that and just go to the conclusions. Okay, so what I told you in this last part is that multifate is a naturally inspired design for multistability. It's based on 2 principles of combinatorial dimerization and autoregulation. It produces a range of behaviors that are similar or analogous to those we see in a lot of natural cell-fate control systems, including multistability, state switching, which I didn't show, irreversible transitions, which I also did not show, and sort of progressive differentiation. It has this beautiful expandability property, which allows you to keep growing the system by adding new transcription factors. And we're really excited about using it as a basis for developing synthetic systems that take advantage of this principle of multicellularity to do things that no single generalist cell type could possibly do. Okay, I also want to say that this circuit design, while it has a lot of attractive features, is certainly not the last word on what circuits can be useful for generating multistability or multicellularity more generally. We think of it as a first step, not the final step. Okay, more generally, just to conclude the whole talk, I think what I tried to convey is really just this one idea that in biology, there's a lot of these many-to-many interaction systems, and that their superpower, or their 2 superpowers, I guess, are 1, kind of enabling computation that can be tailored to cell context, and the other being enabling kind of multistability. And that and also that there's a kind of unexpected feature when we look at the expression profiles of components of these pathways, that they deviate from the hierarchical structure that you might have expected if you thought that the pathways kind of are hierarchically controlled by, or they're, let's say, yeah, just driven by in the same hierarchy that you see for the overall transcriptome, and they're not. So we see examples of these recurring motifs that deviate from that. And I think there's a lot more work to be done there to really understand how those motifs are established and what functions they provide. I tried to acknowledge the key people along the way, and of course there was a lot of other people who contributed to many of these systems and stories. And so let me stop and say thank you for your attention, and I'm happy to answer any questions.